Hello, everyone. Thank you all for joining us today. My name is Claudia Labrador. I am the Global Product Manager of Reagents, Kits, and Assays at Stilla Technologies, and I'll be the moderator of today's session. I'm excited to introduce our first speaker, Laras Pitayu, who is a field applications specialist at Stilla. Laras will be guiding us through the first part of this webinar for research or GMP usage. Nio Analyzer Software streamlines your dPCR data analysis, where we will explore the intuitive features of the Nio Analyzer software for the analysis of dPCR data in research and GMP environments. Throughout this session, we will also discover how Nio Digital PCR can be used for linkage analysis to evaluate DNA or RNA integrity, and learn how to build your high multiplexing panel by color combination with our user-friendly software. We will open the floor for questions at the end of the webinar, and you can submit those under the question tabs on the top right of the menu. With that, I'll hand it over to Lara.
Okay, let me try to share my screen. So I'm going to start this first session. I'm going to introduce the Nio regulatory software, but before, a little introduction about digital PCR and how we do digital PCR with Stilla system and the softwares that are compatible with the Nio system from Stilla. And then we will continue to the regulatory version of the software. So to start, the principle of digital PCR is actually the latest PCR-based DNA detection and quantification that will allow precise quantification without the need of standard curve. So instead of detecting everything in one tube, in digital PCR, the classic quantitative PCR mix will be first partitioned before the amplification takes place. You will have the appearance of the signal that indicates that your encapsulated DNA is detected. And then the analysis will be under this 1D plot form. You have the positive cluster and the negative cluster. The software will then quantify the number of positive droplets compared to the number of total droplets that are generated, or the partition. And then with help of the Poisson statistic, we'll have the result under the form of copy number of target per microliter without the need of absolute quantification. So how we do digital PCR with Stilla? We recently launched the Nio Digital PCR, which is an all-in-one digital PCR system. So to do digital PCR, you will only need one type of consumable. So you prepare your mix and put it in this Ruby chip. One complete run will contain 3 Ruby chips. It can contain up to 48 samples. So it will take around 5 minutes to load your sample. And then you put this 3 chip in the chip plate inside the machine, where the partition and amplification will happen inside the chip. It will take 2 hours or 3 hours according to the program of PCR that you choose. And then it will automatically continue to the reading part for 30 minutes. So the whole workflow will take around 2.5 hours or 3 hours in total. So it's an all-in-one digital PCR system, which is automation ready. It has 7 colors with the possibility of multiplexing that exceeds the 7 colors. My colleagues will explain in the later session. And it can detect up to 768 samples depending on the version of the Nio system that you use. So these are the 3 members of the Nio system. You have the Nio Essentials for people who just start with digital PCR or those who are not having a really high throughput. So you can have one single plate that contains 3 Ruby chips per run. You can run up to 48 samples. And each Ruby chip can contain separate PCR programs. So you can have simultaneously 3 PCR programs with the system. For those who need a higher samples or higher throughput, we have the Nio and the Nio Plus, where you can do up to 4 plates per run for the Nio and up to 8 plates per run for the Nio Plus. And you can do up to 192 samples per run for the Nio and up to 384 samples per run for the Nio Plus. So for the 3 systems, we will need the software, the Nio Reader and the Nio Analyzer to generate and analyze the data. Basically, the Nio reader is a software that is used to control and set up digital PCR experiment on the Nio system. You will generate the Nio data that then will be extracted and analyzed in the Nio Analyzer software in order to calculate the absolute concentration of the target that you are analyzing. And then the Nio Analyzer can also produce config files that can be fed to the Nio Reader software so that your future data will already contain all the configuration requirements in terms of analyze, dot plots, etc. for your future data generation. So this is a brief workflow on how to use the Nio reader. You have the first part, define protocol part, where you will set up the thermocycling condition and define the reading program in terms of exposure time, etc., according to the channels that you will need. And then in the design assay, you can specify the details of your target and putting all the different config files needed for further analysis in the Nio Analyzer. And then in the design experiment, you will combine the protocol and the assay in order to prepare your real experiment. You can upload sample names and also choose the layout of your samples according to your need. And then once the design experiment is ready, you can run the experiment with your real samples. Of course, these 4 steps can be prepared directly from your Nio, but for your comfort, you can also install Nio Reader from your PC, where you can do easily these 3 steps and then generate the design experiment to be loaded directly to your Nio system. So for the Nio Analyzer, again, it's a software that you use to analyze your data. I will present some of the key features of the software. We offer openness, where you can see all your data to the level of every fluorescence in every partitions generated within the droplets. What you see here is actually the picture of one Ruby chamber containing one type of sample in one type of channel. It is famous for its ease of use. Our customers appreciate the user-friendly interface. You have the possibility to visualize in dot plots, polygons, and you can also display the concentration side by side for presentation needs, etc. The Nio Analyzer also provides assay optimization tools such as separability score, which allow the user to evaluate how separated the cluster of the positive and the negative clusters are. This is very useful when you are comparing different PCR conditions for a routine use. You would want to have a separability score that is high. We also have the possibility to have the uncertainty curve display. So you can know the optimal concentration that you need of your target that will give the lowest error possible. Another key features of the Nio Analyzer is that you can connect your 2D plot with the corresponding fluorescent image. What we do here is that we generate a polygon that chooses the specific region within the dot plots. And then we can connect those regions with actual image of the fluorescence within the crystal image. We can also have a fully customizable compensation matrix to overcome spillover fluorescence problem, which is a problem where we have sometimes positive signals from one channel that can be detected slightly in other channel. So we can generate compensation matrix that can actually correct this type of error to assure that you have a correct quantification. And of course, the 2 softwares, Nio Reader and the Nio Analyzers, are available in a regulatory form, so it can be compatible with the FDA regulation 21 CFR Part 11. So the 21 CFR Part 11 from FDA is where you have the need to have the electronic records and electronic signatures for your data. It will allow you to have full traceability of your experiment. It's important for GLP and GMP practices, and it concerns people in industry, in drug development sector, biomanufacturing, or quality control processes. So for the version of the Nio software in 21 CFR Part 11 is fully controlled by user. And then the Nio regulatory software will also form elements that are created, modified, stored, and also secured against further modification, and also traced in detail. It will provide also the functionality of electronic signature for every data generated. So the elements that are included in the regulatory software only differs in one software, which is the NICA data service, which will function as the NICA system account manager and will control the compliance of all the operation that is within respect to the 21 CFR Part 11 regulation. The Nio Reader version and the Nio Analyzer version of the regulatory software will function as the same as I will explain earlier. So the Nio Reader will function as the operating interface, and the Nio Analyzer will function to analyze and to generate data, with also a difference according to the requirement of the 21 CFR Part 11 that I will explain later. So I will first describe the features of the NICA data service. So the NICA data service will actually act like an umbrella of all digital PCR operations under the compliance of 21 CFR Part 11 within the NICA data service, the Nio Reader, and the Nio Analyzer software. It will allow the tracing and also the recording of every operation. So you have what we call the audit trail for every operation of the digital PCR within your Nio system in this regulatory mode. It will also introduce the notion of permission of the user. It means that it can limit the scope of action of different roles in Nio Reader and Nio Analyzer. For example, as a lab manager, you have a wider scope of experiments compared to an operator. You can also specify a personalized role that I will explain in the next slide. So these are the example of the roles that can be managed within the NICA data service. You have different roles in the NICA, in the Nio Reader, and the Nio Analyzer. Every roles that I listed here are accessible for a lab manager, for example, and on operators are limited with roles with asterisks to simply run experiment and generate PDF report. And you can also create a specific and personalized role for other functions within the lab that will be personalized according to the need of the project. And now I will explain the Nio Analyzer regulatory software, some of the features and the functionalities. So how to activate the regulatory version, both for Nio Reader and Nio Analyzer. We will activate the regulatory version by choosing the regulatory mode during installation that will be done by Stilla or a competent person. It will be free of charge for both Nio Reader and Nio Analyzer. So both for these two software, the Nio Reader and the Nio Analyzer, only authorized individuals that are listed within the NICA data service will have access to these two softwares. So user must log in using individual user authentication, username and password, in order to access their data in these two softwares. So this is the workflow example in the Nio Analyzer regulatory software. After the user authentication, you will do the data analysis as your experiment required you. And then instead of continue directly for data extraction, etc., the Nio regulatory version users will need to validate first the sample. For each sample, you need to validate or invalidate according to the criteria that you establish for this specific run. And then you will need to do electronic signature after flagging all the sample in the experiment. There is also a possibility to remove a signature to comply to two-man rules or 4 eyes principle in the GMP settings, because sometimes it requires a data to be reviewed by two persons in order for it to be validated, depending on how it will be applied in your lab. And then you can also create PDF report that will englobes all the different information of the user and software traceability. You can also have experiment and analysis setup information, and also the thermocycling program. In the PDF report, it will also possible to have details of every chamber, every samples. You can also choose to include the audit trail that will list all the different modification from the beginning of data generation to the different step of analysis and modification. So there are several installation options for the Nio regulatory software. The installation option 1 will require, of course, the Nio, the PC that are going to access the data in the Nio, and another PC that will act as a server. So it will create a closed environment within the Nio and the other PCs that are listed to access the Nio. And the installation for the second option, the Active Directory will be generated by using the Nio system as a server. This will require the Nio system to be on all the time so that the NICA data service can always be available for every people who want to access or generate the data from the Nio. And then for the third option of installation, you will only involve a single Nio system. So it's a local Windows setting. So people who generate data and who analyze data will do it only on the Nio system. You won't be able to analyze the data in the software that are outside the Nio system. People can only extract the PDF report as the common PDF file that you can obtain from any other interface. So with this, I end my presentation about the Nio regulatory software. I hope that I cover all the important elements for this session. And now I will leave the floor to Jean Fatien, a senior engineer R&D for Reagent and Assay development. Jean will explain some linkage analysis data and also examples in the HIV study. Thank you all for your attention.
Thank you very much, Laras. Okay. Good afternoon and good morning to all of you. Thank you very much for being here with us. Today, I would like to discuss with you what is linkage analysis and why digital PCR is a great measurement technique. And to do so, I will start with a two-target, two-color example, and I will detail a little bit the computation principles that are needed for this analysis. And then, in the second phase, we'll take a customer case. We'll see the example of a 5-plex assay that was developed by a team at University of Ghent to evaluate HIV integrity. Let's start with the basics. What is linkage analysis? So the situation is I have, let's say, two targets, target A and B, and they can either be on two distinct DNA or RNA molecules. So that would correspond to, for instance, plasmid after restriction, or a fragmented messenger RNA, or a chromosome crossover, etc. Or I can have the two targets present on the exact same DNA molecules. So the two targets are physically linked. And this situation would correspond to an intact plasmid, or a full messenger RNA, a full viral vector genome, etc., etc. So if you want to detect that in digital PCR, it's very easy. You simply need to develop, for instance, here a duplex assay. So you'll have one system, primers and probe, to detect target A. Here I took a blue fluorophore to detect this target, and another system to detect target B, here with a red fluorophore. So what happens during the partition and during the PCR to allow us to detect the linked versus not linked target? Let's do it step by step. Sorry. Okay, so during the partition, the short target A will be encapsulated randomly in the different droplets. After the amplification, I will have a positive signal in blue for some of these droplets. And to estimate the concentration for target A, I will use this formula that is well known to digital PCR users, -1 over partition volume times logarithm of 1 minus positive partitions for target A divided by total number of droplets in the crystal. This ratio is what's going to give me my quantification. So in this example, I have for the positive droplets for target A are simply the droplets that are blue positive. In this case, I have 5 divided by 15 total number of droplets. That gives me an encapsulation rate of 0.33 for target A. Target B will be encapsulated randomly as well. I'm going to use the exact same formula to compute the concentration. And in this case, the positive droplets for target B are the droplets that are red positive. I have 1, 2, and 3 here. 3 divided by 15, I have an encapsulation rate for target B of 0.2. Important to note that since the two encapsulation events, encapsulating target A and encapsulating target B, are independent, the rate of co-encapsulation will simply be the product of the two encapsulation rates. And in this example, it's 1 divided by 15. Okay, now let's see what happens with my linked targets. So this is also just a DNA molecule, so it will also get encapsulated randomly in the various partitions. Using the exact same computations, I will find that my positive droplets for targets linked AB are the double positives. In this example, 3 divided by 15, encapsulation rate of 0.2 for the linked targets. All right. Now, if I have a heterogeneous sample, I know that in my sample I can have the short target A, short target B, but also the linked targets. In this case, the double positive droplets will come from two scenarios. It can either be the encapsulation of the linked targets, but it can also be the co-encapsulation of the two short fragments. So from the quantification that I will derive from these double positive droplets, I actually need to subtract the quantification given by the co-encapsulation of target A and B to find my real quantification for the linked target. And to do this, I need to be able to estimate this rate of co-encapsulation. As I told you before, to do so, I need to have the rate of encapsulation of target A and the rate of encapsulation of target B. The problem here is I cannot derive those rates the way I used to before. I was using just the blue positive droplets divided by total number of droplets to get the encapsulation rate of A. But in this case, that will give me wrong results because I have some droplets that are blue positive, this one for instance, but that does not contain target A. So I cannot assume that all the blue positive droplets are positive for A. This is wrong. What can I do then? Should I just look at the single positive in blue, so blue positive, red negative, and then divide by the total number of droplets? Well, that would also be wrong because in this case, I would have 3 positive droplets, the 3 blue pos red neg, divided by 15, that would give me a rate of 0.2. When actually, if you remember correctly, I should find 0.33. Okay, so let me rearrange a little bit the crystal so we can see things a little bit better. What I'm going to do is use the fact that the encapsulation, 3 events, encapsulating target A, encapsulating target B, and encapsulating the linked target AB are independent. So measuring the ratio of positive droplets over negative droplets for the target A on the whole crystal is actually the same as measuring this ratio on a subset of the crystal that does not contain the other encapsulation events that could be misleading, okay? So if I simply exclude all the red positive droplets from the analysis, then I have non-ambiguous droplets. I have 3 positive droplets for target A divided by a total number of 9 droplets, and that gives me the correct 0.33 rate of encapsulation for target A, okay? And as you can see, I'm using 2 populations, the blue positive, red negative, and the blue negative, red negative. And those are the populations that need to be defined in your experiment in the Nio Analyzer software if you want to perform linkage analysis. Of course, I would measure the encapsulation rate of target B in a similar way, and then I would be able to subtract the co-encapsulation event from the double positive droplets, and I will find the correct quantification for the linked targets. Okay, let's try to sum up what we've seen. So linkage analysis, basically, the situation is simple. I have several targets, and I want to know if they are present on the same nucleic acid molecule or not. That is to say, are they physically linked? And the 3 features that I think are essential and that are brought by digital PCR are first the partition, because that will allow to segregate the different molecules in different partitions. Then the multiple color co-detection. To be able to see the linked targets, you need to be able to detect all the colors that are present on the same molecule at the same time in the partition. And third, the population identification. You need to be able to properly set the correct populations to account for the random co-encapsulation of the separate targets and ensure you do not overestimate the amount of linked targets. Okay? All right. Let's move on now to the customer case. So that was a 5-plex assay for HIV integrity analysis. Very briefly, HIV is a virus with an RNA genome of about 9 kilobases. Today, about 44 million people live with HIV, and that includes 1.3 contaminations last year, 1.3 million contaminations. Unfortunately, a definite cure for HIV has not been found yet, and that comes from the fact that HIV creates reservoir cells. Those are cells that have a complete viral genome integrated in their own genome. So as soon as therapy is interrupted, viral replication can restart. This should be distinguished from cells that may have integrated parts, only fragments of the HIV genome, and which are not able to restart viral replication. And so in order to find a path to finding a cure for HIV would be to be able to eliminate completely those reservoir cells. And to do so, researchers need to be able to properly quantify the size of the reservoir. They need to be able to quantify the amount of intact HIV genome versus total potential fragments. So they need to perform this linkage analysis. So the HIV Cure Research Center team at University of Ghent developed a 5-plex assay for that. They have published the paper on bioRxiv, and it's been accepted recently in clinical chemistry. And so the assay they have developed is this one. It targets 5 different regions of the HIV genome, regions that are relevant for the viral replication. And as you can see, they cover nearly the whole length of the 9 kb genome. The colors used are yellow, blue, red, infrared, and teal. This assay was developed on a different digital PCR platform, but we had the opportunity with them to test it on the Nio system without any modification to the program or to the assay content. If you are interested, you can find our application note on this topic using the following QR code. Very briefly, for the samples, we used genomic DNA from JLAT 10.6 cells. Those are cells that contain 1 copy of intact HIV genome per cell. The extracted DNA was serial diluted 3 times independently, and the concentrations targeted in the digital PCR reaction ranged from 2,000 copies per microliter down to 0.5. In terms of assays, the 5-plex assay was evaluated, of course, but also restriction versions, let's say. So the 4-plex without the infrared, the 3-plex without the yellow, and the single-plex with only the blue targets detected. And of course, if you have only one target, the blue, for instance, you will detect all the potential fragments that contain these targets. So that will be the total HIV quantification, let's say. Whereas as you increase the number of targets, you will be able to distinguish between the short fragments and the longer ones. This was all tested on the Nio Plus system with Ruby chips using 3 replicate wells per dilution point. Okay, I'm going to show you just a couple results from this study. So first, the quantification of the intact genome. So to compute that, we proceeded exactly in the same way that I showed you before with 2 colors, but with 5. So a lot more populations to define, and you've got to compute stepwise the encapsulation rate of the 1 target fragment, then the 2 target fragment, the 3, the 4, until you reach the 5 target fragment. Okay, so here I'm looking at the intact genome quantification from the 5-plex assay. And this is from the 4 first dilution points. This is the average. So of course, with the 5-plex assay, what will come out as intact will be only the fragments that contain the 5 targets. When I'm using the 4-plex, I don't have the infrared signal, so the fragments that will come out will be those ones. And here, because I'm using genomic DNA from cells, I can only have fragmentation happen because of the DNA extraction, because of the DNA handling. But I cannot have rearrangement. So I cannot have a short fragment that would contain the yellow and the teal, but not the middle target. And that's why here, by going from the 5-plex to the 4-plex, I actually get the exact same quantification for my intact genome because I have the two most extreme targets. Then when using the 3-plex, I lose the yellow signal. So in this case, both the fully intact genome and a slightly shorter one will be detected as intact genome because they both have the 3 colors. And in this case, my quantification, as you can see, is a bit higher. So I will overestimate my proportion of intact genome by about 13% in this example. Finally, if I'm using the single-plex assay, I can detect all these different fragments as intact genome. So in this case, I have an overestimation of the intact genome quantification by more than 152%. So as you can see, if you want to evaluate the integrity of a genome, of a gene or a plasmid, etc., it can be very interesting to have multiple targets spread across your genome to make sure that you get the most accurate quantification for your intact fragment. And this is the last result I want to show you, and this is the bias that can arise if you simply take the multiple positive droplets as your quantification for intact fragment. If you do so, and if you do not correct for the co-encapsulation of shorter fragments, you will overestimate your quantification of intact genome. And in this case, the effect will be higher as you go higher in concentration. This is the highest point of the dilution curve. And in this case, I have much more co-encapsulation events. So my overestimation of the intact fragment would be about 14 if I don't use the specific computations I've showed you before. Of course, if you're using very dilute samples, then you have very few co-encapsulation events, so you don't have this problem. Okay. All right. So I told you about the population addition that you need to do in Nio Analyzer, but I did not show you how to do it in the software. And this is actually what my colleague Sana will do. Sana is a senior scientist in the Reagent and Assay development team, and she focuses on developing highly multiplexed assays, and she will show you an example of that just now. Thank you very much for your attention, and Sana, you have the floor.
Thank you, Jean, for the nice introduction, and good afternoon, everyone. So in the last part of this webinar, I will present you what is the standard approach in multiplexing and how you can increase the number of targets by using color combination. And I will also highlight a specific feature within the Nio Analyzer to edit population whenever you're using color combination. So as you probably know, the most straightforward approach is assigning one color to one target. So with this method, and especially using our Nio instruments, as we have 7 channels available for this instrument, you can go up to 7 targets if you are using the standard approach. So here on the right, it's just a schematic where we decided to have 2 targets, the target A in blue and the target A in red. Just next to it, you have a 2D dot plot. So this is the kind of graphs that you will get on the Nio Analyzer as well, and I will show you later. And just to show you how you can visualize the different population on it. So basically, on the 2 axes, you have the fluorescence in red and in blue. And you will find the negative population, so the population which is negative for blue and red right here. And if your targets visualize within your droplets, you will get a population appearing here, which will be positive in blue and negative in red. And this will be the reverse for the target B, as it will be positive for red and negative for blue. So how do you do when you have more than 7 targets that you want to visualize within one assay? So the first thing can be to add more channels to your instruments. But the thing is that we already have the highest number of channels for a digital PCR instrument on the market. So the best solution is not to increase the number of channels, but you can use color combination. And color combination is when instead of using one color to detect one target, you will combine 2 colors to visualize one target. And as you can see on the table right here, and because we have an instrument having 7 channels available, we can go up to 21 targets whenever we are combining the different colors together. And as using color combination can make the analysis more complex, you will see that with our Nio Analyzer software, it will automatically generate accurate and precise data as long as you edit the population and tell the software what will be the color for each population. So very quickly, just to remind you that there are two different approaches whenever you want to perform color combination. The first one will be competing probes. So basically, you will design on the same sequence of DNA two probes. So they will basically hybridize on the same area and they will compete together. And the second approach is to use non-competing probes. So here, the probes will be designed next to each other. So they will not compete for the same sequence of DNA. But at the end, the result is the same. So here we have the 2D dot plot again. And for detecting the target A, which is supposed to be red and blue, you will have a double population, which will be positive for red and for blue here. And this is the same for the target B with two different fluorophores. So before moving to the Nio Analyzer software, where I will show you exactly how to edit a population whenever you're using color combination, I will just introduce quickly the example that I will use to show you the different features. So basically, we are using an assay that has been developed at Stilla, targeting HPV. And we decided to choose 11 HPV subtypes that are linked to cancer. As you can see here, this is the list. And there is also a reference human gene for accurate measurements. So this is the reference. So here you can see in the table the different combination. This assay is already available on the website if you are interested. And don't forget to flash this QR code because you will have access to our technical notes regarding color combination in Crystal Digital PCR with another example, but at least it will give you also the different steps, even if I am presenting right now everything. So let's move to the software right now. So let me share. There it is. So this is not the regulatory version of the software, but the population edition works exactly the same on the two versions of the software. So here you can see the different chambers where we put different template DNA. So in A1, we have the positive control, which is the mix of the 11 HPV subtypes. But then you also have HPV 16, HPV 18, and HPV 31. So you can zoom in and you can have a look at the crystal with the positive droplets. You can change also the different channels and visualize only the droplets that are interesting for you. Then we can move to plot and population, and basically we will have access to the dot plots. So you have different dot plots. You have the 1D, 2D, 3D. So I will show you, for example, the HPV 31 population. Here you can see that you have positive droplets in green and teal, and it's supposed to be the right combination if you remember the huge table that I shared with you right before. We can also have a look at the 2D dot plots, and we can see, as I showed you on the schematic, that this population, which is supposed to be teal and green, appears right here, and it's positive in teal and positive in green. But if you look right here, we can see that there is a population that is already set up for HPV 16 and HPV 18, but there is no population set up for HPV 31. And this is because we will make it together right now. So if you want to edit a population, you will go to the population editor feature. Then you can add a population right here. So it's supposed to be teal green. So let's put teal and green and HPV 31. You can even choose the color you want. So then we will edit the negative droplets. So basically, we will tell the software which droplets should be counted as the population. So they have to be teal and green, but also we have to remove everything that will be negative for these colors. So first we can choose here that the population has to be teal and green. So I will click on teal and green and I will remove yellow, red, and infrared as negative. So we want only the droplets that will be teal and green. And I will keep blue as undefined because remember the blue is the reference. So we want to take into consideration every droplets that are teal and green, even if the blue is here or not. It doesn't matter. That's why it's here undefined. And then automatically, whenever you click on the positive and negative, the software will choose for you the best option for the positive droplets. But you also have to edit the negative droplets. And basically, what will be negative? All the droplets that have absolutely no signal. So I click on blue 0, teal 0, green 0, etc. And everything that it's only blue will also be considered as negative. So I click on it. So we are all set. Then I can click on apply. So now the population should be set, and we can verify that very quick. So I'm on the right chamber with the template HPV31. We do see droplets here. Let's see if we can highlight them whenever I'm clicking. If I look at droplet of interest, you can see that now they are highlighted as purple. It means that this is your population, and this is the droplets that are teal and green. And if you go to the data, so the software will automatically compute the data for you. Now you have already the two populations that were already set up, but this is the new one that we set up. You have the number of droplets counted in your chambers, and it's automatically calculated. You will get the copy per microliter directly for each population. And you can also export the data very quickly, very easily from the software. Let's go back to the presentation. As take-home messages, we would like to emphasize that Stilla technology supports complex assay design. As it has been shown, you can design integrity assays, and you can also design very high multiplex assay for precise target detection, especially when you have a lot of targets. We reached an unmatched multiplexing capability as we have the highest channel capacity in the market right now with these 7 colors. And this allows us to go up to 21 targets whenever we are doing color combination. We also show you that the software is very easy to use, user-friendly, and it can generate very quickly automated data. You have a powerful data visualization as well. So you can visualize the crystal, you can see 2D or 3D dot plots, and you can also customize the reports that you want to generate with your data. And to finish, it's also compatible with the 21 CFR Part 11 regulation from the FDA whenever you're purchasing the Nio Analyzer regulatory. So thank you for your attention. And you can write your question on the chat, and we will take the question right now. Thank you.
Thank you very much, Sanafo, and thank you very much, Laras and Jean, for these very interesting talks. So right now the floor is open to questions. So feel free to tap those in. For the moment, I already have some questions available to start the discussion. So starting with the color combination subject, there's a question about which approach will be preferred, the competing or the non-competing for multiplex color combination, and why?
So the non-competing is the best approach. This is what we prefer to do if we can, because we want to avoid that competition, because it can happen that you have a drop in the efficacy of the hybridization whenever there is competition between the probes. So if we can, it's better to use the non-competing one.
Thank you, Sana. There's another question about the analysis of the linkage for 3 or more targets can be done in the Nio Analyzer, or it has to be performed outside.
Yeah, a very good question. Currently, there is not yet a tool inside the Nio Analyzer software to allow you to do the linkage analysis in 3 or more colors. So you would need to export your data after having correctly defined the populations and use the Excel export to compute that. But of course, if you buy an assay that has been developed by us from our catalog that uses integrity analysis, we have just launched a 4-plex kanamycin resistance gene integrity analysis assay, by the way. Or if you want to develop with us, if you want us to develop a custom assay for integrity analysis, of course, we will provide everything that you need to perform the analysis.
Thank you, Jean. There's another question about color combination. So is it possible to measure more than 2 colors per droplet, hence increasing the multiplexing possibilities?
I don't think this is the best approach. I think it will depend on how many targets you have, because at some point it will be hard to distinguish between, like, if you have three colors and one color which is same for two different targets, I think it's complicated to discriminate from which target it's coming. But yeah, maybe Jean has a better idea.
That's a very good question. That is something that we have never tried before. From a purely theoretical point of view, if you use 2 colors per target with 7 different colors, you can have 21 different populations. If you increase that to 3 colors per target, that will be, let me try that. That would be 35 different populations that you can create, I think. So you can increase the number of the multiplexing that you can reach theoretically, but then the design becomes really challenging. And then if you increase even more, if you go to, let's say, 5, 6 colors per target, then you start to decrease the number of combinations you can do again. If you use 6 colors per target, that's actually the same as using 2 colors per target.
Thank you very much for that question. It is a very interesting one. Now there's another question about, there is any limit in use concentration overall or for a specific population, or can the whole range be used for color multiplexing? And if yes, any implication on higher concentration?
So basically, if there is a limit in the number of copies or the quantity of DNA that we can use whenever we are doing color combination, I would say, but I'm talking under the control of Jean again, it's better not to start with too much DNA, because you will also increase the potential co-encapsulation as well. So I think it will decrease the efficacy first of your assay, and it can be trickier to analyze.
Yeah, that I can complement that. That is a very good question, and we actually have covered that in a paper that has been published and that is available from our website. So what happens when you do color combination is that if you have co-encapsulation of 2 targets, the droplet can become positive for 3 colors, let's say blue, green, red, and you don't know if that comes from the target that has blue, green and the target that has green, red, or if it's the target that has blue, red and the target that has green, red. So you don't know which targets are present. So you need to exclude these droplets from the analysis. If you have very high concentrations of several targets present at the same time in the sample, you will have a lot of co-encapsulation events, so a lot of droplets that you will need to exclude from the analysis. At some point, this will start to decrease the precision that you can have on the quantification of each target. But that really depends on the context of your sample. If you are in oncology and you have an assay where you have a wild-type gene detected and then different mutations that are never all present at the same time in the sample, then what we recommend doing is using one color for the wild type and using the other colors for color combination. That way, even if you are at a very high concentration, you don't need to exclude the wild type droplets from the analysis because they are not ambiguous. So you can keep all the droplets you want for the analysis of each mutation present. And since you can have only 1 or 2 mutations at the same time in your sample, that's okay. You won't have a lot of co-encapsulation events to exclude.
Thank you so much, Jean. So I guess, okay, there's more questions coming in. So there's one question about the presented linkage analysis for HIV genetic integrity that had 4 colors. So the question, I guess, if the linkage analysis is using 4 colors. I guess it was a quick confusion between the kanamycin assay and the linkage analysis example.
Yeah, so the kanamycin assay that we've just added to our catalog is a 4-plex assay, so just 4 colors. For the HIV assay that I presented, we tested up to a 5-color assay. And those 5 colors are actually very useful when you analyze samples, because in those samples, you can have rearrangement of the genome. So you can have all the possible combinations of each color. So being able to distinguish between a 4-color and a 5-color fragment is really important. In the samples that I showed you, where we had genomic DNA, the rearrangements were not possible. So in that case, the difference between detecting 4 or 5 colors was not useful.
Thank you, Jean. I'll be looking at the chat just in case. As we're running out of time, if there's any unanswered questions, we'll follow up via email. So I wanted to really thank again our speakers today for the very interesting insights on how to use the Nio Analyzer software and the regulatory version of both the Nio Analyzer software and the Nio Reader software. If you're interested in learning more about all the all-in-one Nio digital PCR, the GMP environment, how can we help compliance with the software, as well as integrity analysis, color combination, all the subjects that we discussed today, do not hesitate to contact us or to visit us at stillatechnologies.com. Thank you again for your participation today. Bye-bye.